Previously, we learned about how the nervous system functions as a major control center working through somatic and autonomic, and cranial and spinal nerve reflexes to protect the body and maintain homeostasis. The nervous system conducted these activities in milliseconds with pinpoint delivery to specific effectors. Working along with the nervous system, the endocrine system functions to maintain homeostasis, but it works in a completely different manner. The endocrine system releases chemical messengers called hormones into the blood or lymph. The hormones circulate about contacting the body's tissue cells. When they find their specific targets, they initiate a sequence of events that makes the target cell carry out a particular function. Because the endocrine system uses the circulation to distribute its messages, its action is slower in onset, and its effects are more prolonged compared to the nervous system responses. The endocrine system consists of the major endocrine glands plus endocrine cells spread throughout the body in almost all of the body's organs. No matter what the size or location, endocrine glands have a couple of characteristics in common. One, they are ductless, so they have to dump their secretions directly into the blood or lymph. And because of this, they are too highly vascularized, meaning that they have extensive blood supply. The location of the major endocrine glands are shown in the above picture. Unlike the digestive, cardiovascular, respiratory, and other organ systems, the major endocrine glands are spread throughout the body and usually have no physical connection with other endocrine organs. As we shall see, there is a lot of overlap between endocrine glands and other organs from different systems. Above are listed an example of the overlap between major endocrine organs, tissues, and cells with other organ systems. For example, the hypothalamus is part of the brain, yet it functions also as a very important endocrine gland. The thymus is part of the lymphatic system where T lymphocytes receive their programming. Yet the thymus secretes the hormone thymosin, which plays a regulatory role between brain and immune system, as well as sustaining certain aspects of immune system function. The bulk of the pancreas produces digestive enzymes as an exocrine product, but islands of endocrine cells within the gland produce the hormones insulin and glucagon, which help regulate blood glucose levels. The ovaries and testes produce the gametes or sex cells, but they also secrete steroid sex hormones. The list goes on. In this unit, we will cover the functions of the major endocrine glands and cover the function of some of the accessory cells and tissues when we study the other organ systems they reside in. For now, it would be very useful for you to learn the information on this slide. It shows the location and the hormone products of the major endocrine organs, glands, or cells. As mentioned previously, the endocrine and nervous systems maintain homeostasis by regulating the activities of other organ systems. The nervous system is fast, site-specific, and its actions are short-lived. Neurons work by either inhibiting or stimulating cells or organs. The endocrine system broadcasts its instructions via blood-borne hormones referred to functionally as chemical messengers, which circulate throughout the body. Endocrine responses are slower in onset and more prolonged in action. Hormones contact every cell in body, but only certain cells with receptors specific for the hormone will respond to it. These cells/organs are called target cells for the particular hormone. There are three major chemical classes that all hormones fall into. These are 1 amino acid derivatives, 2 peptide hormones, and 3 lipid derivatives. Amino acid hormones are small molecules derived from amino acids. They have different side groups than the amino acids comprising proteins. Of the amino acid hormones, there are 2 subdivisions. Those hormones derived from the amino acid tyrosine and those derived from tryptophan. The derivatives of tyrosine include the catecholamines, epinephrine, norepinephrine, and dopamine. You know from previous presentations on the nervous system that all three of these are neurotransmitters. They also can be secreted into the bloodstream where they function as hormones. Another extremely important tyrosine derivative are the thyroid hormones. We shall see that many cells of the body have receptors for thyroid hormone, and its roles are many and varied. The amino acid tryptophan is what makes people sleepy when they eat turkey, or drink a warm glass of milk before going to bed to help them sleep. Tryptophan is needed to make the pineal hormone, melatonin, which regulates sleep-wake cycles. Peptide hormones are small chains of amino acids, or small proteins, usually under 200 amino acids long. Some of these bear carbohydrate residues, making them glycoproteins. These include several pituitary hormones, erythropoietin, made by the kidney, and inhibin, which plays a key role in reproductive physiology. The short polypeptides and small proteins are the largest group of hormones and include hormones from the hypothalamus, pituitary gland, heart, digestive tract, pancreas, parathyroid gland, C cells, adipose tissue, and lymphatic system. The third category are the lipid derivatives. Some are derived from a membrane lipid called arachidonic acid. Prostaglandin is probably the most commonly known hormone belonging to this group. The other type of lipid derivative are the steroid hormones. All of these are derived from cholesterol. They include the sex hormones, as well as calcitriol-active vitamin D3 from the kidney, and corticosteroids such as cortisol and aldosterone from the adrenal gland. Different hormones make their targets work in different ways. The main way this is accomplished is by regulating movement of substances in and out of cells. In other words, changing membrane permeability to different substances such as glucose, insulin, fatty acids, amino acids, secondary active transport, ions. Aldosterone regulates sodium/potassium levels. Iodide is necessary to make active thyroid hormones, water antidiuretic hormone, and they can even regulate gene expression by working of specific genes. You don't have to memorize all the steps illustrated in the diagram above, but there are a couple of important concepts that you should be aware of. First, the chemical nature of the hormone is going to affect how the hormone causes changes in its target cells. Amino acid derivatives, peptides, and small proteins are not soluble in the lipid bilayer of the plasma membrane. Therefore, they must bind to a receptor protein embedded in the plasma membrane. Hormone receptor binding activates a series of intracellular molecules that result in the formation of cyclic AMP or release of calcium ions. These substances act as second messengers. Remember, the first messenger was the hormone that couldn't diffuse through the lipid membrane and so can't act directly. In the case of CAMP, ion channels may be opened up to allow the inward flow of specific ions, or it could activate enzymes that make the cell do something else. In the case of calcium acting as a second messenger, hormone R, receptor binding leads to the activation of a membrane lipid enzyme, which results in the formation of protein kinase C. PKC opens calcium ion channels in the plasma membrane, allowing calcium to flow into the cell. In addition, many cells have specialized smooth endoplasmic reticulum that store and concentrate calcium ions. Recall the sarcoplasmic reticulum of muscle cells. Binding of certain hormones ultimately result in the release of stored calcium ions from the SER called calcisomes. In either case, calcium alters the activity of specific enzymes and channels, or it binds to the intracellular regulatory protein calmodulin. Once calcium binds to calmodulin, it activates enzymes that amplify the cellular response. The lipid-derived hormones are much easier to understand. because of their solubility in lipid membranes. This class or hormones do not have to bind to a membrane receptor. The example above shows the steps involved when steroid hormones activate their targets. First, the steroid hormone diffuses through the plasma membrane and binds with an intracellular receptor. The receptor may be in the cytoplasm or in the nucleoplasm. Either way, the receptor hormone complex binds a hormone response element, a specific DNA sequence corresponding to the start of a particular gene. The binding initiates transcription of the gene to mRNA. The mRNA leaves the nucleus through the nuclear pores, becomes associated with ribosomes, which directs protein synthesis. The protein product may be another hormone to be secreted, or it may alter the activity of the target cell. As mentioned in the introduction, the nervous and endocrine systems are the two major homeostatic regulatory systems of the body. They control the activity of almost everything else. Not all that long ago, it was discovered that the nervous and endocrine systems are both structurally and functionally related. And this realization led to the formation of a new branch of medical science called neuroendocrinology. It turns out that the pituitary gland, once thought to be the master gland, is the key to the relationship between the two organ systems. The pituitary gland hangs from the base of the brain, specifically the hypothalamus. It has a dual embryological origin and consists of two lobes. The adenohypophysis anterior lobe is formed from oral ectoderm as an outpocketing of the mouth called Rathke's pouch. The neurohypophysis posterior lobe develops from the floor of the diencephalon from the area that will become the hypothalamus. Rathke's pouch pinches off the remainder of what will become the digestive tube and becomes associated with the brain. Recall that the pituitary gland sits in the sella turcica of the sphenoid bone. The prefix adeno means gland. There are 6 distinct anterior pituitary hormones, all proteins. Of the 6, 4 are tropins or tropic hormones, which means that they regulate the secretory action of other endocrine glands. These include thyroid-stimulating hormone TSH, which targets the thyroid gland and stimulates the release of thyroxine. Adrenocorticotropic hormone ACTH stimulates hormonal release from the adrenal cortex. Follicle-stimulating hormone FSH stimulates production of ova and secretion of estrogen in females and sperm production in the testes in males. Luteinizing hormone LH works with FSH to stimulate estrogen production, ovulation, formation of the corpus luteum, and secretion of progesterone. In males, the equivalent hormone is interstitial cell stimulating hormone ICSH. It stimulates secretion of testosterone by the testes. The remaining hormones work directly on their targets. Growth hormone HGH. Human growth hormone regulates growth and metabolism by targeting bone. Melanocyte-stimulating hormone MSH stimulates melanocytes to increase synthesis on melanin pigment in fish, amphibians, and reptiles. But this is not an important function of MSH in humans. Interestingly, MSH in humans and other mammals is a CNS neurotransmitter involved in the control of appetite. However, in Addison's disease, there is an increased pigmentation of the skin due to excess MSH. Prolactin PRL stimulates synthesis and production of milk. All anterior pituitary hormones, except growth hormone, exert their effects on their targets via CAM. Neurons of the hypothalamus secrete releasing and inhibiting hormones into the primary capillary plexus at the base of the hypothalamus. Blood with hormones drains into hypophyseal portal veins, which pass down the anterior part of the infundibulum. Portal veins form a secondary capillary plexus in the anterior pituitary. Hormones enter sinusoids, which causes secretion of pituitary hormones. The hormones enter the systemic circulation and bind with receptors on their target cells. Above is a list of the hypothalamic hormones that regulate the activity of the adrenal hypophysis. The synthesis and release of most hormones is regulated by some type of negative feedback system. Some internal or external stimulus triggers hormone secretion. As hormone levels rise, they cause target organ effects, which then inhibit further hormone release. As a result, blood levels of many hormones vary only within a narrow range. The picture above shows three types of endocrine feedback control patterns of the affecting the adrenal hypophysis. In the general pattern, the hypothalamus secretes a releasing hormone which causes the cells of the anterior lobe to release a tropic hormone, which in turn causes a peripheral endocrine gland to release its hormone. As the levels of the second hormone rise, this feeds back to both the hypothalamus to stop the releasing hormone and the pituitary to stop the release of the tropic hormone. There are a couple of pattern variations. For example, secretion of GH is regulated by two hypothalamic hormones with antagonistic effects. Growth H, hormone-releasing hormone GHRH stimulates GH release from the adrenal hypophysis, whereas growth hormone-inhibiting hormone GHIH inhibits it. GHIH release is triggered by the feedback of GH. Rising levels of GH also feed back to inhibit its own release. Prolactin functions to stimulate milk production by the mammary glands. Prolactin release is controlled primarily by an inhibitory hormone, prolactin-inhibiting hormone PIH, which turns out to be dopamine. PIH prevents prolactin secretion. Decreased PIH secretion leads to a surge in PRL release. In females, prolactin levels rise and fall in rhythm with estrogen blood levels. Estrogen stimulates prolactin release, both directly and indirectly. In pregnant women, PRL blood levels rise dramatically toward the end of pregnancy, and milk production begins. After birth, the infant suckling stimulates release of prolactin-releasing factors in the mother, promoting continued milk production. The posterior pituitary is called the neurohypophysis and consists of the median eminence of the hypothalamus, the hypophyseal stalk, and the posterior pituitary or pars nervosa. Two hormones are released from the neurohypophysis. Antidiuretic hormone ADH is formed in the supraoptic nucleus of the hypothalamus. ADH or vasopressin regulates urine volume and blood pressure. The second hormone is oxytocin. This hormone is involved in promoting labor contractions and the milk let down reflex in females, and contraction of smooth muscle lining the ductus deferens and prostate in males. Oxytocin is synthesized in the paraventricular nucleus of the hypothalamus. As it turns out, the pars nervosa or posterior lobe of the pituitary does not synthesize the hormones. The posterior lobe consists of unmyelinated nerve fibers, pituocytes, which are neuroglial cells, and swellings easily visible with the light microscope called herring bodies. Herring bodies are actually expanded axonal endings of the hypothalamic neurons coming from either the supraoptic nucleus or paraventricular nucleus, and contain granules of either ADH or OT. ADH and OT are synthesized in the hypothalamus, transported down the axons, and stored in the axon terminals located in the pars nervosa. The red arrows in the diagram and micrograph of the pars nervosa indicate the swollen ends of the axon terminals contain granules of ADH or OT. This figure shows how the hypothalamus controls the pituitary gland. Notice that the adrenal medulla is under direct control of the nervous system via the sympathetic division of the ANS. The remainder of the hormones are controlled indirectly via releasing and inhibiting factors from the hypothalamus. The posterior pituitary releases ADH or oxytocin via direct release from the axon terminals herring bodies of the hypothalamus. The thyroid gland is bilobed and lies over the larynx. The lobes are connected by a narrow bridge of tissue called the isthmus and extend laterally to the common carotid arteries. Try palpating your thyroid gland. Physicians do this routinely as part of a physical examination. Also notice the massive blood supply going to the thyroid gland. This is necessary to bring regulatory messages via TSH to the thyroid and to take away the functional, active thyroid hormones. Because of its embryological origin, the thyroid gland contains three different functional systems located together in a single gland. 1. The main thyroid gland. 2. The parathyroid glands, and parafollicular or C cells. The primary structural and function unit of the main thyroid gland is the thyroid follicle. It consists of a ring of typically simple cuboidal epithelial cells surrounding an amorphous center consisting of colloid. Between the follicles are the extrafollicular or parafollicular, also known as C cells. These cells respond to elevated levels of calcium in the plasma by releasing the hormone calcitonin. On the backside of the thyroid gland, there are a variable number of separate parathyroid glands. The typical condition is to have 2 parathyroid glands per lobe, but some people have more or fewer. The parathyroid glands respond to low levels of calcium in the blood. Let's examine the main thyroid gland first. Shown in the upper right-hand corner is an isolated thyroid follicle. Note the capillaries surrounding it. The lumen contains colloid, which will be converted to thyroglobulin, the stored form of thyroid hormone after iodine has been added to the colloid. When TSH is present, it binds to TSH-sensitive ion pumps, which are specific for iodide. The iodide is removed from the bloodstream and enters the follicular cells via their basal surface. The iodide diffuses through the cytoplasm of the follicular cell to the plasma membrane. The plasma membrane contains an enzyme called thyroid peroxidase, which attaches the iodide to a chunk of colloid-forming thyroglobulin. The follicular cells endocytose the thyroglobulin. Lysosomes fuse with the vesicle and digest the thyroglobulin into its 2 major active forms found in the tissues and bloodstream. T3 and T4 tri- and tetraiodothyronine equal thyroxine. Notice that T3 and T4 are small hormones, amino acid derivative. Thus they bind to transport plasma proteins so that they are not filtered by the kidneys and lost in the urine. The thyroid hormones are gradually released and diffuse into the peripheral tissues located throughout the body. Thyroid hormones are important for running many metabolic processes efficiently to support homeostasis. Thyroid hormones affect almost every cell in the body. Thyroid hormones regulate basal metabolic rate (BMR), protein synthesis, ATP production, and energy turnover, body temperature, proper function of the nervous system, and are important in the fetus and children for normal patterns of growth and development. The concentration of thyroid hormones in the blood and in tissue cells is held in check through negative feedback mechanisms. As circulating levels of thyroid hormones begin to decline, the blood bathing the hypothalamus and adrenal hypophysis is deficient in thyroid hormones. This triggers the hypothalamus to secrete TRH, thyroid releasing hormone, into the capillary plexus, where it is delivered to the adenohypophysis. This causes the secretion of TSH into the bloodstream, which circulates throughout the body, ultimately arriving to the thyroid gland. TSH causes the thyroid gland to secrete thyroid hormones into the bloodstream. As the circulating levels of thyroid hormones increase, They feed back negatively, causing the hypothalamus to slow down or stop secretion of TRH, and the pituitary to slow down or stop secretion of TSH. This results in thyroid hormone levels returning to optimal levels. If the thyroid gland or adenohypophysis or hypothalamus are not working properly, then homeostatic imbalances in the level of circulating thyroid hormones can result. In the case of too little thyroid hormone or hypothyroidism, in adults results in many metabolic and nervous system disturbances such as slowed basal metabolic rate, which causes weight gain, excessively dry skin, thinning hair due to inadequate protein synthesis, slowed reflexes, lethargy, and depression. Not uncommonly, people suffering from hypothyroidism are misdiagnosed as being clinically depressed and are prescribed antidepressants. These, however, do not reverse the condition. A simple blood test for circulating levels of TSH would indicate that the individual is depressed because they are hypothyroid. This condition is easy to fix by treating the patient with thyroid hormone pills. If you were treating a patient that you suspected might be hypothyroid, would you expect the level of TSH to be high or low? Myxedema is an extreme example of full-blown hypothyroid syndrome in adults. The term myxedema means mucus swelling. It results from accumulation of mucopolysaccharides in the connective tissues. The condition can be reversed by administering thyroid hormone. It can be considered the adult counterpart of fetal cretinism because of the slowed reflexes and mental function, and depression. Hypothyroidism can result during fetal development with disastrous consequences. Lack of adequate levels of thyroid hormone causes cretinism in which the individual does not meet developmental landmarks. The individual is dwarfed due to metabolic disturbances and mentally retarded because thyroid hormone is necessary for the proper development of the nervous system. The most common cause of fetal hypothyroidism is because the mother was not obtaining adequate levels of iodine in her diet. Therefore, the fetus's thyroid gland could not make active thyroid hormone, resulting in severe somatic and nervous system deficits. The picture above is of a patient suffering from Hashimoto's disease or chronic thyroiditis. Notice the massive size of the thyroid gland. In general, an enlarged thyroid gland is known as a goiter, but there may be several different causes for the enlargement. The following information is excerpted from Wikipedia and the Mayo Clinic. Hashimoto's thyroiditis is an autoimmune disease that very often results in hypothyroidism with bouts of hypothyroidism. Physiologically, antibodies against thyroid peroxidase and or thyroglobulin cause gradual destruction of follicles in the thyroid gland. Accordingly, the disease can be detected clinically by looking for these antibodies in the blood. It is also characterized by invasion of the thyroid tissue by leukocytes, mainly T lymphocytes. It is associated with non-Hodgkin lymphoma. Symptoms of Hashimoto's thyroiditis include myxedematous psychosis, weight gain, depression, mania, sensitivity to heat and cold, paresthesia, fatigue, panic attacks, bradycardia, tachycardia, high cholesterol, reactive hypoglycemia, constipation, migraines, muscle weakness, cramps, memory loss, infertility, and hair loss. The thyroid gland may become firm, large, and lobulated in Hashimoto's thyroiditis, but changes in the thyroid can also be non-palpable. Enlargement of the thyroid is due to lymphocytic infiltration and fibrosis rather than tissue hypertrophy. Hypothyroidism results when too much thyroid hormone is secreted. Symptoms include increased BMR, weight loss, tachycardia, very rapid heart rate at rest, and irritability. Graves' disease is an autoimmune disorder that results in hypothyroidism. As you know, All our body cells have markers on their surfaces letting the immune system know that they are self, so that antibodies and lymphocytes don't destroy them. In autoimmune equals self-immune diseases, this recognition system breaks down and the immune system begins making antibodies that attack the person's own cells. In Graves' disease, the body makes antibodies that mimic TSH. Therefore, the thyroid gland responds by oversecreting thyroid hormone. This results in the development of a diffuse goiter, enlarged thyroid gland, weight loss, rapid heartbeat, fatigue, muscle weakness, frequent bowel activity, restlessness, heat sensitivity, hand tremors, and increased sweating. National Women's Health Information Center et al. 2006. In addition, certain individuals with Graves' disease display symptoms associated to this form of hypothyroidism, including infiltrative ophthalmopathy, bulging eyes, thyroid acropathy, periosteal bone formation, and pertibial dermopathy, abnormal deposition of hyaluronic acid. Hypothyroidism can be treated with propylthiouracil, a compound that blocks binding of thyroid hormone. In general, an enlarged thyroid gland is called a goiter, but there may be different reasons for the enlargement, and the person may be either hyperthyroid or hypothyroid. In the case of hypothyroid goiters, the gland is unable to produce active thyroid hormone. Most commonly, this is caused by a lack of iodine in the diet. Because there is little thyroxine in the bloodstream, there is no negative feedback, so the hypothalamus keeps secreting TRH, and the adenohypophysis keeps secreting TSH. The follicles become congested with colloid that cannot be converted to thyroglobulin because of the lack of iodine. So the colloid just keeps accumulating until the follicles become tremendously enlarged. The patient is hypothyroid, and this type of goiter is called an endemic or colloidal goiter. Conversely, goiters may be caused by oversecretion of TRH or TSH, which causes the follicular cells to keep synthesizing and secreting thyroid hormone. In this case, the goiter contains small amounts of colloid because it is being converted to thyroid hormone and secreted, but the follicular cells become tall and columnar and are densely packed. Thus, the goiter is a result of enlarged follicle cells as they try to meet the demand placed on them. The other two components of the thyroid gland are involved with regulating plasma calcium levels. The parafollicular or C cells, indicated by the white arrow in the histology slide of the thyroid gland, secrete the hormone calcitonin in direct response to elevated plasma calcium levels. There are typically 4 parathyroid glands attached to the posterior surface of the thyroid. Each gland contains 2 types of cells. The principal or chief secretes parathyroid hormone PTH in direct response to lower than optimal levels of plasma calcium. Notice that blood calcium levels are held within a very narrow range, optimally between 9-10 mg/100 milliliters. Plasma calcium ion homeostasis is essential for proper nerve impulse transmission, muscle contraction, blood clotting, and secretion. When plasma calcium levels get too high, the C cells release calcitonin, lowers plasma calcium levels by increasing excretion from the intestines and kidneys and stimulating osteoblasts to lay down bone. Recall that osteoblasts are immature bone cells that secrete bone matrix. Calcium salts are used to make the bone matrix hard, so calcium is taken out of the bloodstream and incorporated into new bone matrix. PTH is secreted in response to low levels of plasma calcium. It increases plasma calcium and magnesium levels and decreases phosphate levels by decreasing excretion of calcium by the kidneys and intestine through activation of vitamin D calcitriol, and increases the activity of osteoclasts. Recall that osteoclasts are bone cells that reabsorb bone matrix. In the process, calcium salts are released into the bloodstream and normal calcium levels are restored. Notice also that abnormal levels of calcium in the blood affect the permeability of sodium in cell membranes. When blood calcium levels are too high, sodium permeability decreases, which makes the cells less sensitive and responsive to stimuli. Recall that cells respond to stimuli by changing sodium permeability. If sodium can't enter the cell easily, then the cell becomes less able to respond to stimuli. Conversely, if blood calcium levels are too low, sodium permeability of the cell membrane increases, bring cells closer to threshold so that they are irritable or too sensitive to stimuli. Hyperparathyroidism is a rare disorder that is usually caused by a tumor of one or more parathyroid glands. The result of hyperparathyroidism is elevated serum calcium levels called hypercalcemia. Under the constant influence of PTH, calcium is leached from the bones. The bones soften and can become deformed. The bone matrix is replaced by fibrous connective tissue. Other effects include depression of nervous system function, which leads to abnormal reflexes and weakness of skeletal muscles. Also, because of the high plasma calcium levels, formation of kidney stones is common. Hypoparathyroidism is due to deficiency of PTH, so the individual is hypocalcemic. This results in an increased excitability of neurons and accounts for the classical symptoms of tetany, loss of sensation, muscle twitches, convulsion, respiratory paralysis, and death. The pineal gland is also called the mind's eye because it is sensitive to light from the retina in mammals, but contains photoreceptors in lower vertebrates. Descartes and others dubbed it the seat of the soul because of its position between the hemispheres, and most likely because they had no clue what it did. Today, the pineal can be considered part of several different organ systems. The nervous system, because it is part of the brain, visual system, because it contains photoreceptors in lower vertebrates used for detecting light levels, the endocrine system, because it secretes the hormone melatonin, and the reproductive system, because in seasonally breeding animals it affects the gonads. The cells of the pineal are called pinealocytes. They synthesize melatonin, which is liberated during darkness. When light strikes the retina, melatonin release is inhibited. This relationship between light and pineal aids with our diurnal rhythms. We feel sleepy at night because as the light levels decrease, the melatonin levels increase. Some animals hibernate during the long, dark, cold winter months. This saves them a lot of energy because their metabolism is lower to the point where they don't need to eat. Heart and breathing rates are very slow, as well as other metabolic functions. Although people don't hibernate, there is a condition known as seasonal affective disorder, SAD. SAD is a common disorder in people living in higher latitudes. This is because during the winter, there are very few hours of daylight, so melatonin is being released. The elevated levels of melatonin make people lethargic, depressed, and leads to carbohydrate craving obesity. People crave carbohydrates because the brain's levels of serotonin have declined, and carbohydrates are used to boost its level. Thus, people gain about 10 pounds over the winter and then lose it in the summer when the days are longer. People who suffer with SAD can be treated using light therapy. A couple of hours under the lights and melatonin levels decline. Melatonin also affects the gonads in seasonally breeding animals. During the winter, the gonads of both male and female animals involute and greatly decrease in size. This is because gonads require a lot of energy input to make gametes. Therefore, when there is no need for them during the non-breeding season, melatonin works with the reproductive system to reabsorb the gonads. In the springtime, longer daylight hours suppress melatonin release and the gonads and gametes begin to grow. The adrenal glands, also known as suprarenal glands because they sit on top of the kidneys, also have a dual embryological origin. Therefore, the adrenal glands consist of two structurally and functionally different regions. The adrenal cortex is the outer region. It is derived from the mesoderm near the developing urogenital structures. Mesoderm is the middle germ layer that develops during early embryonic life. The adrenal cortex produces steroid hormones, which are essential for life because they regulate electrolyte balance and glucose metabolism. The cortex is subdivided into zones. Each zone produces different hormones. The outermost zone, located just underneath the connective tissue capsule of the gland, is called the zona glomerulosa. Glomerulus is a general term for ball or sphere, so the term zona glomerulosa refers to the sphere-like arrangement of cells. The zona glomerulosa produces mineralocorticoids. In humans, the major mineralocorticoid is aldosterone. Aldosterone regulates sodium and potassium levels in the blood by acting on the renal tubules, gastric mucosa, sweat, and salivary glands. Aldosterone is secreted in response to decreased sodium in the blood, decreased blood pressure, increased blood potassium level. Too much aldosterone increases sodium levels and decreases potassium levels in the blood, results in water retention, increased blood pressure, and if potassium levels fall too low, faulty conduction of nerve impulses and inability for muscle fibers to contract adequately. Note: most release of aldosterone from the zona glomerulosa is not via ACTH from the pituitary gland. Aldosterone production and secretion is regulated by renin produced by the kidney. Aldosterone secretion occurs in response to angiotensin II. More on this when we discuss renal physiology. The zona fasciculata zone of Styx gets its name because the cells in this region are lined up in more or less parallel rows. The cells of the zona fasciculata secrete mainly glucocorticoids. The major human glucocorticoid is cortisol, hydrocortisone, and cortisone. The glucocorticoids affect carbohydrate metabolism mainly and have a lesser effect on protein and lipid metabolism. However, they do promote fat deposition in certain regions of the body, like the belly. You may have used hydrocortisone cream to stop the itching of a rash or an inflammatory response. Glucocorticoids are given to patients receiving organ transplants and to people with allergies or arthritis to decrease inflammation or suppress rejection of a transplanted organ. Glucocorticoid release is controlled by ACTH secreted by the pituitary gland and CRH by the hypothalamus. The innermost layer of the adrenal cortex has its cells arranged in a net-like pattern, and thus is called the zona reticularis. The zona reticularis produces sex steroids. Since the gonads are the major source of these hormones, it is not clear just what the function of the adrenal sex steroid is. It has been reported that the adrenal androgens male sex steroids are important for libido in females and appropriate sexual behavior in both males and females. Oversecretion in children result in the precocious development of the genitalia and appearance of pubic hair before puberty. The adrenal androgens have little effect in adult men. However, oversecretion in adult women causes increased muscle mass, blood cell formation, hair growth, and changes in libido. As with all steroid hormones, the adrenocortical hormones are built on a backbone of cholesterol. Release of cortisol is regulated via the hypothalamus-pituitary axis. When cortisol is needed, the hypothalamus releases CRH, which causes the adenohypophysis to secrete ACTH. ACTH works on the zona fasciculata to cause the release of cortisol. Notice that nerve impulses from higher brain centers can influence the secretion of cortisol because of the connections between the cerebrum and the hypothalamus. Therefore, When people are subjected to chronic stressful situations, they have elevated levels of cortisol in the blood. There are two common diseases that result from abnormal regulation or secretion of glucocorticoids, mainly cortisol. Addison's disease results from undersecretion of glucocorticoids, whereas Cushing's disease is the result of oversecretion of glucocorticoids. Addison's disease results from damage to the adrenal cortex. The damage causes the cortex to produce less of its hormones. The damage may be several factors, including autoimmune disease in which the adrenal cortex is attacked by antibodies, infections such as tuberculosis, HIV or fungal infections, hemorrhage, tumors, or the use of anticoagulants. Cortisol deficiency produces disturbances in carbohydrate, fat, and protein metabolism and results in severe insulin sensitivity. In the absence of cortisol, insufficient carbohydrate is formed from protein, which results in low levels of blood glucose, known as hypoglycemia. The hypoglycemia results in muscle weakness, due in part to deficient neuromuscular function. People with Addison's also have difficulty maintaining normal blood pressure, hypotension, and have reduced resistance to infection. One of the most interesting symptoms of Addison's disease is increased skin pigmentation. Decreased cortisol blood levels result in increased pituitary ACTH production, causes excess release of MSH, which produces the hyperpigmentation of skin and mucous membranes. Cushing's disease results from excessive levels of corticosteroids. This leads to chronic high blood pressure, hypertension, and high blood glucose levels, hyperglycemia, progressive obesity, altered distribution of adipose tissue to the abdomen, and development of moon face and buffalo hump. In addition, the skeletal muscles lose bulk and weaken, and the skin thins and bruises easily. The bruising that occurs on the abdomen as a result of increased fat distribution to that area resembles stretch marks. The inner part of the adrenal glands is called the medulla. The cells in this area, known as chromaffin cells, come from a ridge of neuroectoderm called the neural crest, which is the same origin as the sympathetic nervous system. Instead of becoming neurons, these cells become endocrine cells. The cells of the adrenal medulla produce two hormones, epinephrine and norepinephrine, same as the neurotransmitters of the sympathetic nervous system. These play a major role along with the sympathetic nervous system in the fight versus flight reactions. As alluded to previously when discussing the role of cortisol in chronic stress, the adrenal gland has two different stress responses depending on how long the stressor lasts. For example, short-term stress, as occurs in fight versus flight reactions, is mediated through preganglionic sympathetic fibers that cause the chromaffin cells of the medulla to release epinephrine and norepinephrine. However, prolonged stress is mediated through CRH from the hypothalamus, causing increased release of ACTH, which causes the adrenal cortex to secrete both mineralocorticoids from the zona glomerulosa and glucocorticoids, mainly cortisol, from the zona fasciculata. Increased levels of cortisol increase blood glucose levels, but also suppress immune function. This is why many students get sick around finals. As you've probably noticed, there is a fairly common theme of dual origin when discussing the major endocrine organs like the pituitary, thyroid, and adrenal glands. The pancreas is no different. The bulk of the pancreas, about 90% of its mass, is involved with secretion of digestive enzymes, which is an exocrine type of secretion. The digestive enzymes are synthesized and then released into ducts that fuse with more ducts, finally forming the main pancreatic duct that delivers digestive enzymes to the small intestine where they work to digest our meal. The endocrine part of the pancreas makes up the remaining 10%. The endocrine part of the pancreas is in the form islands of endocrine cells that are dispersed throughout the gland. These clumps of cells are called the islets of Langerhans. There are 4 different types of endocrine cells found in the islets. Alpha cells secrete the hormone glucagon, which increases blood glucose level. Beta cells secrete insulin, which decreases BIOL, blood glucose. Delta cells secrete somatostatin, which inhibits secretion of insulin and glucagon. And F cells secrete pancreatic polypeptide, which helps to regulate release of digestive enzymes. In this section, we will be concerned only with the activities of glucagon and insulin in regulating blood glucose levels. The endocrine pancreas responds directly to circulating levels of blood glucose. The hypothalamus and pituitary gland are not involved. After a meal, the level of glucose in our bloodstream increases. This stimulates the beta cells to release insulin. Insulin stimulates glucose uptake by the tissue cells, and it stimulates the liver to store glucose as glycogen. Consequently, blood glucose level falls back into the normal range. However, between meals, our blood glucose levels begin to drop. The decrease in blood glucose causes the alpha cells to release glucagon, which acts on the stored glycogen in the liver by breaking bonds between the glucose monomers. This releases glucose into the bloodstream and levels rise back into the normal range. This table summarizes the effects and symptoms of an individual suffering from diabetes mellitus. The word mellitus means honey. It was the sweet smell of the urine from patients suffering with diabetes mellitus that gave the disease its name. There are 2 main types of diabetes mellitus, type 1 and type 2. In type 1 diabetes, formerly known as insulin-dependent diabetes, the onset of symptoms appears suddenly and usually strike before the individual reaches the age of 15. Current research suggests that diabetes is an autoimmune disorder involving genes located on several chromosomes. Type 1 diabetes leads to long-term effects such as lipidemia, increased lipids and cholesterol in the bloodstream, resulting in vascular and circulation problems, which can lead to atherosclerosis, strokes, heart attacks, renal failure, gangrene in the extremities, and diabetic retinopathy, blindness. Type 2 diabetes, formerly called non-insulin-dependent diabetes mellitus, comes on gradually with age and weight gain. Most people suffering from type 2 diabetes produce insulin, but the insulin receptors on cell plasma membranes are unable to respond to it. The disease results are the same as people with type 1 diabetes. In the table above, the liver, adipocytes, and muscle are not able to import glucose and use it for production of ATP. Therefore, glucose levels in the blood are elevated, hyperglycemia. The glucose levels are so high in the blood that when the blood is filtered by the kidneys, the renal tubule cells' glucose transporters become saturated, so large amounts of glucose spill over and is lost in the urine. This condition is called glycosuria. The high levels of glucose in the filtrate osmotically pull fluid out of the blood into the filtrate, which increases the volume of the urine. This condition is called polyuria, much urine. As a result, the person is dehydrated and always thirsty, so they drink water constantly, polydipsia, and eat carbohydrates, polyphagia. The liver breaks down glycogen to release glucose into the bloodstream in a process called glycogenolysis. Since the body's cells cannot take in the glucose, different types of macromolecules are broken down to be used as a substrate for making ATP. The proteins actin and myosin in skeletal muscle are broken down and used to make glucose from non-carbohydrate molecules in a process called gluconeogenesis. Adipose tissue releases lipids, resulting in high fat levels in the blood called lipidemia. Production of ketone bodies leads to a drop in blood pH called ketoacidosis, and large amounts of ketones in the urine, ketonuria, are excreted. This causes the respiratory system to increase respiration rate hypernia in order to release more carbon dioxide in an attempt to lower the production of carbonic acid. This is a means of respiratory compensation to reverse the acidosis. The diagram above summarizes 3 different ways that endocrine organs receive the stimulus to produce and release their hormone product. V is called humoral because an ion or molecule in the bloodstream directly stimulates the endocrine gland. Examples include maintenance of calcium ion homeostasis by parthormone and calcitonin. The second is stimulation directly from the nervous system. The example shown above is illustrating release of epinephrine and norepinephrine from the adrenal medulla is through direct stimulation by preganglionic sympathetic neurons. The third mechanism is via hormones, in which the hypothalamus secretes hormones that stimulate the anterior pituitary gland, causing it to secrete tropic hormones that target other endocrine glands.